All right. Good afternoon, everyone. My name is Jose Moran Mirabal. I'm a professor in the Department of Chemistry and Chemical Biology and the scientific director of the Center for Advanced Light Microscopy. And it is my pleasure to welcome you to this last installment of our CALM BIMR seminar series about microscopy, building microenvironments with light, and so on and so forth with all the applications. And so today we are fortunate to welcome to this seminar series Dr. Angelo Accardo from the Technical University of Delft in the Netherlands. And Dr. Accardo is a collaborator and good friend of mine, and so it's a double pleasure to have him here today. I'll give a brief synopsis of Dr. Accardo's trajectory, just so that everyone gets acquainted. So Angelo got his master's degree in electronic engineering from the University of Rome, La Sapienza in Italy in 2008, and his PhD in biomedical engineering from the University Magna Grecia of Catanzaro, Italy, in collaboration with the European Synchrotron Radiation Facility in Grenoble, France. And after a postdoc at the Italian Institute of Technology in Genova, Italy, he joined the Laboratory for Analysis and Architecture of Systems in the La CNRS in Toulouse, France. And so this is where I met Angelo, and we started a very nice collaboration. So he worked there as a research associate until 2019, when he joined Delft University of Technology as a tenure-track assistant professor of soft micro nanosystem technology for life sciences and biology within the Department of Precision and Microsystems Engineering. And so the work that he's going to talk to us about is his work at TU Delft, which is pushing the boundaries of nanofabrication and the study of biomechanics and cellular signaling at the nanoscale. So Angelo, welcome. It's a pleasure having you here.
Thanks, Jose. So this one is always giving this heat glitch. See. All right. So gracias, Jose, for the nice introduction. Thank you, Alex, as well for this very nice invitation. I'm really happy to be here, as Jose said, not just to strengthen the collaboration that I have with Jose, but also to catch up with a very good friend of mine that I met in the beautiful landscape of La Ville Rose in Toulouse. And where I had the opportunity also to collaborate and to work with some of his talented PhD students, Kevin, Sam, and Muhannad Wabi, who is over there. And yeah, so this afternoon, I will show you essentially how it is possible to use light to shape 3D microenvironments, and in particular, how to use these microenvironments for cell biology applications. Before doing that, let me spend a few words about where I come from, where maybe, as you might have recognized from my accent, I'm not Dutch, I'm Italian, and I was born in Rome. But this is where I ended up in 2019. This is an aerial view of part of the campus of TU Delft, also known as Delft University of Technology. In particular, here you can see the building of Aula, where PhD graduation takes place. This one here is the Faculty of Mechanical Engineering, where I'm actually working. This is the Faculty of Electrical Engineering. Then we have the Faculty of Applied Sciences, Civil Engineering. And then I will show you which other faculties we have. And this one here is our very nice library. Let's put it a bit more at the even less zoomed scale. Where is TU Delft? Well, TU Delft is in the Netherlands, and believe it or not, it is located in the city of Delft, which is a very nice little town in the southern parts of the Netherlands, and is part of the western region of the Netherlands, where here you can see Amsterdam, Leiden, The Hague with the international court. Then there is Delft and Rotterdam. So it forms a kind of an L. And as you can imagine, there is no scale bar, which is very bad for an engineer, right? We want always numbers and scale bars. Unfortunately, there is no scale bar here, but Netherlands is a very small country. So each of these cities can be reached within 40 minutes max, okay, when there is no big traffic jam. Which is a point of interest, because especially in my field of research where we combine microfabrication, cell biology, imaging, it is important to bring samples, and especially cells, as fast as possible between each institute. So that makes our life easier. The Netherlands is famous for a lot of things. Well, for sure, for the beautiful tulips, which are growing and flourishing in the spring period, but also for the windmills, the tilted houses of Amsterdam, but also other cities in the Netherlands feature these. But the thing about which the Dutch are most proud of is this one, so-called the croquette. After 3 years, I didn't understand yet which are the ingredients of this. And as an Italian, you know, my culinary threshold is pretty high, so I'm not really interested in that. I tried just once. Was enough. I lost 7 kilos after arriving in the Netherlands. But then again, because I started to cook myself. All right. So this is an overview of the faculties of TU Delft. We have 8 beautiful faculties. Of course, as the name says, we are a technical university, so we don't have faculties of law, for instance, or let's say letters. We have a faculty of civil engineering, applied sciences, industrial design, 3ME. The 3M is because it's the faculty of mechanical, maritime, and materials engineering, where I belong, and the faculty of architecture, aerospace engineering, electrical engineering, and then technology, policy, and management. So TU Delft is a pretty well established university in the Netherlands, but also worldwide. We are proud to say that in the Netherlands, especially in the field of engineering and technology, we are the best. Not in that bad, always of engineering and technology in Europe and worldwide, where in particular, we train our students to become, of course, either engineers, so they go to work to companies like ASML or Philips and so on. But some of them are brave enough to undertake an academic career, and then after the master, they start a PhD. It's not working anymore. Yes. So these are some numbers. We have the size of our, TU Delft is the oldest university in the Netherlands, founded in 1847. And actually, we have more or less 30,000 undergrad students, and we have a PhD population of around 3,000 students with staff, including scientific and supporting staff of around 6.5K people. And we have a very decent output in terms of research publications, which was around 7K in 2021. This is a very nice picture of my faculty, of the faculty of mechanical, maritime, and materials engineering in the evening. A faculty which is composed by 7 different departments. And among these, we find departments related to material science, process and energy, biomechanical engineering, maritime technology, cognitive robotics, and last but not least, precision microsystems engineering, which is the department where I belong to. A department which, it's like a matryoshka, faculty, departments, and then inside the departments, sections. We have 5 different sections in our department, ranging from the so-called modeling guys, which are working on structural optimization and mechanics, dynamics of micro nanosystems, including also quantum nanoscience. Then we have professors working on macro scale devices like mechatronic system design, optics for technology, and last but not least, micro nano engineering, which is the section where I belong. So, before coming to TU Delft, well, as Jose said, I got my master in 2008, and I started my PhD in 2009. But I didn't start to work immediately on these 3D engineered microenvironments. I was always working on technology for biology, but from a different perspective. Particularly during my PhD, I was working on super hydrophobic surfaces which were inspired by nature, by the lotus leaves, like this one here. And in particular, I was studying the mechanisms of droplet coalescence and proteins related to neurodegenerative diseases. PhD partly at the European facility. In order to analyze the mechanisms of this protein aggregation and protein conformational change. That was until the first part of my postdoc in Genova. And then one day, I went to attend a seminar of the head of the neuroscience department, and he started to show all these beautiful, colorful, confocal images of cells and these dendrites, and I said, whoa, very nice. So I said, okay. I asked him, have you ever tried to just grow your cells on something which is a bit less flat than a Petri dish? Then he said, no. Why should I? My approach is very cheap, very easy, very reproducible. And I said, but it's not biomimetic at all. I mean, we don't have a Petri dish in our brain, right? Or at least we hope so. So this is when I started to make use of similar topographies that I was using for creating super hydrophobic surfaces, but this time to culture cells by using conventional manufacturing approaches such as optical lithography, plasma reactive ion etching. And then this brought me to La Ville Rose in Toulouse, where I discovered the mesmerizing world of 3D fabrication, two-photon polymerization in particular. So where I started to study these cells in even more biomimetic configurations. All right. Now, before diving into some of the recent research results of my group, let me spend a few but very important basic concepts. Let's just refresh them. We are human beings. We are organisms, and we are made of unit blocks. We are made of organ systems, which are made of organs, which in turn are made of tissues, and which are ultimately made of the smallest unit blocks, which are cells. Now, in such context, a very important concept is the one of the cell microenvironment. What is a cell microenvironment? Well, a cell microenvironment is any region of space in our body, which is composed by two ingredients, cells, and then a natural surrounding scaffold, which is the guy here in yellow, which is the extracellular matrix. The function of the extracellular matrix is to provide mechanical and biochemical cues to the cells so that they can perform their functions properly. And there is a lot of interest in trying to replicate this extracellular matrix. That's the reason why during the last 2 decades, we witnessed an evolution from 2D cell culture models towards 3D cell culture models. What is a 2D cell culture model? Well, the 2D cell culture model is the typical glass slide approach. What happens? Cell biologists take their cells, they put them in a plastic box, the Petri dish, or on a glass slide, they let them grow and see what happens. Well, I'm making it a bit simple, but just in summary, that's what it is. As I said, there's an approach which is easy to use, reproducible, cheap, but not biomimetic, because it leads in most of the cases to the formation of unrealistic cell monolayers, 2D monolayers, which do not follow the three-dimensional spatial configuration that we find for cells in our body. So that's why during the last, as I said, 2 decades more or less, a new generation of scientists, a bit in between a microfabrication engineer and a cell biologist, started to develop 3D engineered architectures to foster this biomimetic cell colonization by playing on different parameters such as geometrical features, mechanical cues, biochemical functionalization, and so on. Now, in this context, there are 3 terms which must be kept in mind: mechanobiology, durotaxis, and curvotaxis. Mechanobiology is a very recent field of research which is in between mechanical engineering and biology. And the main aim of mechanobiology, like the name says, is to understand how topographic, physical, mechanical cues can influence cell fate, cellular behavior. And linked to this, we find durotaxis and curvotaxis, which are words of Greek origin, which are essentially related respectively to the ability of cells to recognize the mechanical or topographic cues of a 2D or 3D structure, and according to these cues, regulate their behavior in terms of adhesion, proliferation, differentiation, and so on. So the mission of my group, in a few words, is to engineer multi-scale biomimetic microenvironments addressing 3 different kinds of application. We find fundamental mechanobiology applications, the development of in vitro disease and in vitro treatment models, and in a longer-term perspective, tissue engineering applications. At the current stage, my group, which is, well, pretty young because it has been established 3 years ago, well, with less actually because I was alone at the beginning. My first PhD student arrived after 9 months or so, is currently focusing its attention on the development of engineered microenvironments dedicated to 3 types of tissue: brain, which is the core activity of the group; brain cancer, also known as glioblastoma; and bone. And these gentlemen and gentlemen here are the PhD students and postdocs working in the group, which are of course supported also by master bachelor students, which of course spend a bit a shorter amount of time in the lab compared to the very long master trajectory here at McMaster, as far as understood. In TU Delft, master project takes maximum 1 year, of which 3 months of literature survey research plan and 9 months of actual project. Of course, we don't kick them out at the 12th month, but we try to let them graduate in time. Or at least we hope, we try, we hope. All right, now let me start with the brain. When we talk about engineered neuronal microenvironments, so we can discriminate among different manufacturing strategies. We can have based strategies, self-assembly manufacturing strategies. We can have 2D scaffolds. We can have 3D scaffolds. So in this first combination, we find techniques such as optical lithography, soft lithography, nano-imprint techniques, which can allow you to create 2D, 2.5D structures. And in this other category, you can use, for instance, a self-assembly of microbeads or fibers like graphene sheets, graphene fibers, in order to influence the behavior of neuronal cells or neuronal stem cells. But then if you want to go on, if you want to go 3D, let's say, you have to use other techniques. In particular, in the design-based approaches, we find manufacturing techniques such as two-photon polymerization, bioprinting, stereolithography. If you don't have access to these very fancy and sometimes very expensive setups, you can rely also on other self-assembly strategies, including the use of electrospinning, which is a very simple and cheap device, by means of which it is possible to create aligned nanofibers, but with less control on the geometry of the architecture, or the self-assembly of porous hydrogels. And then there is a third category, which is the cell biology based category. It is the category where you find all those approaches which lead to the formation of tissue-like structures, such as neurospheroids, or even more complex architectures like this one here, a neural organoid. In such case, you often make use of ECM-like gels, like matrigel. So you can actually get very complex structures, but one of the main limitations of this approach, first of all, there is a lot of batch-to-batch variability in these gels, like matrigel. And in most cases, in these very large structures, you rapidly develop what is called a necrotic core. So essentially, the inner part of the tissue-like structure starts to literally die because of lack of nutrients and oxygen. And this is not really nice when you want to do long-term drug screening assays. So now, if we focus our attention on 3D printing of cellular microenvironments, the state of the art tells us that most of the approaches are including techniques such as extrusion printing, inkjet printing, selective laser sintering, projection stereolithography, which are great approaches. For instance, using bioprinting or extrusion printing, you have access to a wide library of biocompatible materials, which is very important for 3D cell microenvironment. But these techniques also share some common drawbacks. For instance, the impossibility to create true 3D freestanding structures without the presence of sacrificial supports, and the limited resolution, which is usually ranging between tens to hundreds of microns. In order to overcome this limitation, during the past 5-6 years, I focused my attention on the development of microfabrication paradigms involving two-photon polymerization. Two-photon polymerization is based on the use of infrared laser sources, femtosecond infrared laser sources, and of photosensitive materials, liquid materials, which are transparent to the infrared radiation and very absorptive in the UV one. By exploiting a phenomenon which is called two-photon absorption, by means of which two infrared extremely fast photons are absorbed almost simultaneously by the material in a very confined volume, it is possible to create 3D freestanding structures without the need of sacrificial support, because in this case, it is the non-exposed material to support actually the structure, with a feature resolution that can go down to 200 nanometers, which makes the 2pp the technique with the highest feature resolution achievable. Now, I started to work on 2pp when I arrived in Toulouse in 2016. And in particular, I started to work on the development of neuronal scaffolds made of different kinds of materials. So you have to know that when it comes to 2pp, most of the materials are very stiff. Very stiff, it means with a Young's modulus in the range of gigapascal. In our body, just the bone has that Young's modulus, but not the brain, okay? Even when there is Alzheimer, which stiffen the brain tissue, it doesn't get that stiff. Yeah, okay, too bad. We had to rely on this kind of material and we were curious to see how neuronal cells will develop within this material. I will come back to the Young's modulus, to the intrinsic Young's modulus in the following slides. But just to show you how everything started. So everything started with a very simple 3D wood pile architecture that you can see here, where we cultured neuronal cells that were able actually to colonize the 3D architecture efficiently, both on the external shell, but also in the inner core of the scaffold. This is actually a light sheet fluorescence microscopy 3D reconstruction of the scaffold, where you can actually recognize the cells colonizing the inner beams. One of the most interesting things is that by using the high resolution of 2 pp, I was able also to decorate the cylinders of this wood pile structure with some nanogratings, which acted as guiding pathways for the alignment of neuritic extensions. Very nice proof of concept. But again, the limitation here was the material and the fact that we were using structures which were still pretty thick. It was 20 micron diameter here. So then I started to wonder, okay, let's try to use something which is a bit more biomimetic. And that is why when I started to develop my own hydrogels. And in particular, biocompatible hydrogels, such as polyethyl glycol diacrylate, PEGDA, which is an FDA-approved material. And I combined it with a photoinitiator called IRGA-Q819, which actually allows the triggering of the 2-photon absorption phenomenon required to perform 2-photon polymerization. And in this way, I was able to obtain wood pile structures with a feature resolution very close to the one of the optimal IPDIP, that is the name of this material here, optimal IPDIP 2-photon polymerized material. The difference here is that PEGDA has a Young's modulus which is in the range of hundreds of kilopascal. It's way lower than IPDIP. In this way, it was possible to obtain much more well-ramified neuronal networks with the presence of multiple neuritic extensions per single cell, thing that I could not observe in presence of the IPDIP material. So that is an example of how the softness of the material plays actually a direct role on the evolution of the neuronal network. Also, the material was very interesting from an optical point of view. You must know indeed that whenever you use a 3D cell microenvironments, you will use at some stage a 3D monofluorescence microscopy to track some markers, differentiation markers, proliferation markers, adhesion markers, and so on. If you have a material which is intrinsically fluorescent, then this messes up a bit your work because it will overlap with the fluorescent signal coming from the staining of your cells. In this case, our material, PEGDA, had an intrinsic fluorescence which was more or less 100 times lower than the one of IPDIP, which is the material that we used in this first publication. And this allowed us, as you can see here, to obtain the extremely high resolution 3D monofluorescence reconstruction obtained by 2-photon confocal microscopy in particular. But here you can see in green the beta tubulin neuronal marker, in red the F-actin microfilaments, and in cyan the nuclei. But we are a faculty of, well, when I arrived in TU Delft, we are a faculty of mechanical engineering. So we are also interested in probing the mechanical properties of this material. And this is what we did in collaboration with the International Iberian Nanotechnology Laboratory in Portugal, where by using nano-indenting techniques, we characterized the mechanical properties of 2-photon polymerized PEGDA. And the most interesting thing that we found out was that the 2-photon polymerized PEGDA has a dual stiffness regime, or let's say areas. The inner core, which features a Young's modulus in the range of 1 megapascal. And then the outer layer, which is just 2-3 microns thick, a softer Young's modulus in the range of 100 to 200 kilopascal, which is very interesting because that is the part with which cells are in contact and that the cells sense. And you must know that stiffness plays a role also in terms of stem cell differentiation. among the other things. And then, yeah, I met Jose, and then we started to wonder, okay, nice scaffolds, but let's functionalize them even further, maybe with a coating which is fully natural, like cellulose, and in particular, the very nice cellulose nanocrystals with which Jose is working. And the Muhammad, we worked together in LAS, and we started to develop the polyethyl glycol deacrylate scaffolds, functionalized with different concentrations of CNC, of which you can see the typical dimension over here. And we tested them in presence of mammalian cells to see, among the other things, if there was a correlation between the concentration of the CNCs and the colonization mechanism, colonization efficiencies of the cells. And long story short, by performing a multi-technique characterization involving confocal microscopy as well as scanning electron microscopy, we saw a correlation between the percentage of the CNC coating and the ability of the mammalian cells, in this case PC3 cells, to adhere and colonize the scaffold, which makes it very interesting because PEGDA by itself is bioinert. It's biocompatible, but bioinert. So it's CNCs provide a fully natural biochemical coating for the culture of mammalian cells. Okay, arriving at you, Delft. Okay, let me be frank. Whenever you work with hydrogels and with soft materials with 2PP, it's a mess. It's a mess because they start to collapse, start to swell, start to delaminate. It's not really the easiest material to work with. So with my very first PhD student, Ahmed Sharaf, that you can see over here, we started to wonder, do we really need a soft material for letting neuronal cells think that they are in a soft environment, or can we trick them somehow? So then we started to work with the Biomedical Primate Research Center, the largest primate research center in Europe, in Rijswijk, 10 minutes from my apartment, where they have access to tissues coming from biopsy of primate models, rhesus macaque in particular. And we focused our attention on these beautiful guys here, or ladies, which are called microglia. Microglia are a part of the immune defense system of the brain. They are the scavengers of the brain. When they do not work properly, they can lead to neurological disorders, or they can contribute, let's say, like Alzheimer's or Parkinson's disease. How do they work? In a healthy brain, a microglia displays a so-called ramified phenotype, ramified morphology. When there is a foreign body, like this one here, the microglia surround the foreign body, and they transit from a resting ramified phenotype to a shrunk, non-ramified inflammatory phenotype. Then they phagocytose the foreign body, and then they go back to the resting phenotype. Now, in a healthy brain, as I said, the typical configuration of microglia, this is an immunofluorescence image of a microglia in vivo, so in the tissue, is the ramified one. When you work with primary cells in general, they're very delicate, okay? They're not like cell lines, so they're very picky. And the thing is that when you culture them in a plastic dish, they don't like that. Not at all, not one bit. They immediately develop a shrunk inflammatory phenotype or morphology, which is not ideal if you want to develop drug screening applications. Okay, we look at this microglia, and then we had a look at them at the SEM on 70 gigapascal stiff glass light. And as we were expecting, we observed a few ramified phenotype, few ramified microglia, but mostly amoeboid ones, shrunk ones. And then by 2-photon polymerization, we started to create extremely fine, extremely accurate nanopillar carpets, where in particular we exploited the concept of effective shear modulus. That's the trick. The effective shear modulus, which is represented by this formula over here, is the shear modulus sensed by a cell when it is crawling over a carpet of compliant nanostructures. So when these nanostructures move, even if the material itself, which in this case was IP-deep, the 1.8 gigapascal material of the first example that I showed you, the cells actually feel it much softer because it's not bulk. That is why it's called effective shear modulus. And that depends, of course, on the coverage of the nanopillars and on their aspect ratio. Longer, well, taller, thinner, lower effective shear modulus, which can go down in this case to a few hundreds kilopascal, very close to the rigidity of the brain. And that was the result. The microglia like that, and they developed a very well defined ramified phenotype over 15 days of culture. And here we have also to stress out the fact that this was the result not only of the effective shear modulus, but also of the employed topography. These are 200 nanometer diameter pillars, dimension which is in the range of dimension of the fibers which compose the extracellular matrix in the brain. So we could not disentangle these two, all right? So we think that both effects, topography and mechanical and effective shear modulus, played a role in the guiding the morphology of the microglia. Now, another project of which I'm very proud of is related to brain cancer, in particular to 3D engineer the brain cancer microenvironments for proton radiobiology. Brain cancer is a tremendous disease. I don't have to tell you that, with a very dismal prognosis, 13.5% survival rate in the Netherlands. Now, with brain cancer, you have three options normally: surgery, chemotherapy, X-ray therapy. Surgery cannot be always employed, especially if the mass is big and is in the core. Chemotherapy has a lot of side effects. And in conventional X-ray therapy, X-rays, they kill the cancer cells, but they also travel through the brain. And they also kill the healthy cells around. No good. One alternative is proton beam therapy, where proton beams, compared to X-ray beams, can be accelerated and decelerated due to particle matter interaction to a specific volume where is the tumor, where they release the full energy, and without damaging too much the surrounding healthy cells. End of the story? Not at all, because proton beam therapy is way more recent compared to X-ray therapy, okay? And is also way more expensive. And the combination of these two leads to the fact that much less is known in terms of proton glioblastoma cell interaction. Glioblastoma is brain cancer cell. And you cannot perform these tests systematically on animal models or biopsies. So what we thought, okay, again, let's take inspiration from nature and let's engineer the glioblastoma microenvironments. How? Well, first of all, we looked at immunofluorescence images of the brain and literature. And from literature, it is known that brain cancer cells proliferate and cluster around blood vessels. So the idea here was to use two-photon polymerization to reproduce this microvascular architecture, and then to develop a co-culture model where we would have endothelial cells, which are the building blocks of blood vessel, and brain cancer cells. After that, expose the 3D engineered microenvironment to proton beams and assess the difference between 2D and 3D models in order to create a kind of benchmark tool for proton radiobiology. thanks to the great effort of Kes Akolavala. And actually, the work that I'm going to show you is from a master student. So he developed this within 9 months, and is now, of course, a researcher in my group, because we always keep the good ones. Developed a first version 1.0 of these scaffolds, pretty minimalistic, I will say, still, where we cultured only glioblastoma cells coming from a cell line, so non-patient derived, but commercially available, where they developed a very nice 3D biomimetic network compared to the conventional 2D cell monolith that you have on flat pedestals made of the same material. Then we exposed these 3D and 2D models to protons, and we assessed the level of DNA damage within the cells in 3D and 2D configuration. How? By using a marker called gamma H2AX, which are these green spots here, which essentially are related to the points where there are breaks in the DNA, so where there is DNA damage induced by the radiation. By counting these spots, you can give an estimate of the level of DNA damage. And what you observed was that 2D models compared to 3D models exposed to different radiation suffered more, showed more DNA damage foci, which are this one here, compared to the 3D model, which was in line with what was observed when comparing real tissues with a 2D cell culture model. So in other words, we developed a platform that could be used as a benchmark tool for proper radiobiology, but without using biopsies or models, at least at first stage. But as I said, that this model is still, this geometry is still, at least in my eyes, still very simple. Oh, there is also this part I forgot. We also looked in this work at the morphology of glioblastoma cells, and we observed for instance, we observed that the average nuclear area was way larger in 2D compared to 3D, where we observed more globular morphology, like it happens in vivo. Same for the cytoskeleton. And we also looked at the length of the intercellular protrusions. And in particular, we saw that in 3D, these were developing longer protrusions, which is also in line with what is observed in an in vivo configuration. Yes, spherical morphology, as I said. And the other thing was that, interestingly, the nucleus in 2D, let's call them 2D cells, was rather localized in the upper region of the cytoskeleton surface, while in 3D, the nucleus was embedded more within the cytoskeletal envelope, which is a proof of how cells readapt their cytoskeleton when they go through a meander of microstructures which try to mimic a hypothetic extracellular matrix. We are very interested in glioblastoma, and Nastaran Barin, this lady over here, who was also one of my master students and now a PhD student shared between TU Delft and Erasmus Medical Center, is working on that. In particular, one of the main differences with Kaz's work is that she's working with cells coming from patients. So we take a biopsy from a real patient, we mechanically and enzymatically dissociate the tissue into cells, and then we culture the cells on 3D scaffolds. In particular, in this case, mostly to look at the morphology of the cells and to see if we can spot similarities with cells in their native environment. And this is actually the latest, actually not the latest one, but very recent work, which was accepted a couple of weeks ago. For instance, we looked at the nuclear area, all right? So this is from the 2D model, and this is from the 3D model. And as you can see, the nuclear area, as you observed for the glioblastoma cell line of Kaz, is in average way lower than the 2D model. And then the thing which we did in plus was characterization of glioma cells from in their native environment, which is also in line with the previously reported literature. Then another aspect that we studied, the formation of so-called tumor microtubes. Tumor microtubes in primary glioma cells are cellular protrusions which are directly involved in cellular malignancies and passiveness. These cellular protrusions are often observed in vivo. For instance, this is in a mouse brain. But when you develop 3D glioblastoma models using, for instance, spheroids, it is not possible to induce their formation. Using our model, we were able to spot several of these microtubes, which were analyzed both by using immunofluorescence microscopy, but also using a scanning electron microscopy, where in particular, we could easily identify the presence of both micro and even nanotubes, as you can see here, and where, again, like for the nuclear area, the quantitative characterization, in particular, in this case, for what concerns the protrusion width, which, as you can see, in the 3D model is lower in average than the 2D model, and again, in line with previously reported the values of microtube protrusions for cells in vivo. But of course, all these studies are done in static conditions. Cells move. And one of the main challenges is to be able to track the movement of cells and see how do they behave, right? So this is a scaffold which was colonized over 16 hours, where by tracking the cells and by comparing the migration speed in 2D and 3D, we observed, and we expect this result, that in average, the level of speed of patient-derived glioblastoma cells is twice slower compared to 2D. Why? Simply because they encounter more structures, so they are stopped compared to the completely free 2D. We also looked at the stability of our cellular network. For instance, here you can see a tumor microtube in 3D and in 2D. And we observed for how long this tumor microtube was preserved, where here you can see that in average in 3D, the tumor microtube were for around 4 hours, or less than. In two things. Either the glioblastoma network is more stable in 3D, or the fact that the cells are more rapidly moving to the has an influence also on the stability of the network in 2D, something that we have still to further investigate. And then the last thing that I want to show you related to this work is actually how do these cells migrate? Because normally glioblastoma cells in the brain, they migrate along blood vessel. Remember, they cluster and proliferate along blood vessel. And here you can see a video of how this happens in vivo. And in vitro, in our 3D models, we were able to see in our 3D model, we were able to see similar behavior. So a directional movement along the beams of the scaffold, compared to a much more chaotic and isotropic migration behavior on the 2D plate. All right, so that's the end of the story. And let me also spend just a few moments to acknowledge properly all of my collaborators. This is part of my group because some of them joined more recently during our last outing in the beautiful canals of Delft, which are really nice from April on. Don't come before April, it rains a lot. And of course, my collaborators from the Biomedical Primate Research Center for the microglia project, collaborators from Vrije Universiteit of Amsterdam for the autism spectrum disorder project, LUMC for the glioblastoma, and Erasmus MC as well. Of course, Jose, Mohamed, and Eduardo for the CNC related project. My very good friends, Christophe and Laurent, with whom I worked for 3 wonderful years in Toulouse before coming to TU Delft. The people from the Holland Proton Therapy Center, and of course, the collaborators from Vilnius University and the INL in Portugal, the funding agencies, but most of all, your attention, and thanks for coming here today.
Thank you, Angelo. Very good presentation. I'd like to open the floor for questions now and encourage students, first of all, to ask questions.
First of all, wonderful presentation, very insightful. So just wondering about the stereolithography. So how stable are the structures when subjected to a biological environment?
That depends on the material that you're employing. In these examples that I showed is more about two-photon polymerization rather than stereolithography. So if you're using acrylate polymers with intrinsic Young's modulus in the gigapascal range, they are stable until a certain extent, because when you are culturing, for instance, what you observed with the preostoblasts, after 21 days of culture, the preostoblasts, they proliferate, of course, and they actually start to deform the structure. And we want them to deform the structure because we want them to sense this auxetic or non-auxetic behavior, okay? So it depends a bit on the type of cells that you have, how much invasive they are. For instance, brain cancer cells are very invasive, but mostly depends on the geometry of the structure and the flexural stiffness of the structure and the material which is applied.
How does, you said this is influenced by curvataxis. So at what point does the cell begin to behave and respect the structure, then go on to have a different approach?
So if I well understood your question, until which extent the cell follows the topography, or the topography is modified by the cell?
Yeah, so you said at one point it functions and grows within the structure that's made, but then once it reaches a certain point, it's grown.
Oh, yes. Yes, that depends simply on cell density. So, yeah, the group is more than a single. So when the group start to form also a matrix, by means of which it is connected to other cells which are way further from the cell A, then this collective behavior promotes the deformation of the structure.
There is a question online, and the question is, will these cell culture techniques become available for general use in the near future? And if not, what would it take to bring them?
For general use, what does it?
Commercially available.
Oh. Short answer, no, because 2pp is not really well suited for high throughput. The more resolution you want, the slower is the longer is the fabrication time. There are some developments in using parallel. It's called parallel infrared beamlets. So that means essentially instead of having a single beam, infrared, the femtosecond infrared beam, to split this beam into a matrix of beamlets so that you can actually photopolymerize at the same time a different region, several different regions of your architecture. But this is a technology which is under development. I do not see it commercialized before the next 5 years.
So a lot of the structures that you showed are either wood piles or there was one where it was more of a dome. How well does that translate to the matrices in the brain, and how much does it translate into how it actually is in a environment, or sorry, a bio? Why do you choose the structure?
So the wood pile was just a matter of simplicity. It was just the first design that I wanted to test, and you start always with simple. Doesn't make sense to start too complex. For the glioblastoma engineered microenvironment, for instance, we started with straight substructures, but having the same diameter of the blood vessel, but straight, because they were more mechanically stable, and we didn't want to, again, make it too complex. The dome configuration so has these curved beams, which are more reminiscent of the microvessels, and the dome is because of imaging reasons, because we want to make sure to image without any shadowing effect the colonization of cells at several layers in the outer shell of the architecture and in the inner core of the architecture. So when you have a dome and then you have an upright microscope, which is flashing light, if you have something which is square like this, like the first 3D wood pile, you have many more shadowing effect and many less photons which go back to the detector compared to something which is like pyramidal or dome shaped. More complex, what do you mean?
So that's sort of like the simple structure of like file,
The microvessel is an example. With Peter, the gentleman working on autism spectrum disorder 3D engineered models, we are looking at so-called Voronoi designs. The Voronoi designs are pretty random structures, and which are a bit more reminiscent of the natural extracellular matrix, because essentially the geometry of the extracellular matrix is a pure mess. Doesn't follow any specific geometry, but it is also true that the other tissues of our body are extremely well aligned. If you think about the spinal cord, another thing that we are I'm working on as a project proposal, there you need very well aligned guidelines, because the gray matter of the spinal cord is formed by axons completely parallel. In that sense, then you need something which is extremely ordered and not random. Thanks. Thank you.
So with the structure and design, I saw you use the activated tag as the structure, right? And my question is like, how have you ever tried to, so the cells have the channel response to this structure with different responses. So have you tried this patterning of soft, soft regions and harder regions, see if that affects the cell migration or differentiation? Research like that.
That's a great question, and it's actually a bit along the line of what I showed for this multi-material two-photon polymerization. So in that case, we use materials with different Young's moduli. I mean, with different, 2-3 order of magnitudes difference. But probably what you're talking about is to have gradients of the same material. In that case, it is possible. It is more challenging from a mechanical point of view, mechanical stability of the structure. And as a mechanical engineers, we are always very concerned with that. But it is possible by creating fabrication paradigms using different laser exposures or different what we call writing speed within the same architecture, so that you can, if you write slower, you expose more, and you photopolymerize, you cross-link more your material, and then it becomes stiffer. Same thing if you use like a lower power, of course, you cross-link less and it becomes softer. Yeah, the main problem there is the mechanical stability, which is it's a matter of a trade-off. But yes, gradients, if you think about the brain, the cortex, our brain is made by layers, and these layers are populated by neurons and glial cells. So it will be a dream to have a structure which features stiffer regions on the top, softer regions at the top, so that the IPS-derived neuronal progenitor cells can differentiate into glial cells, and the stiffer region at the bottom for the neuronal cells.
Yeah, that'll be really.
No, yes, I agree. Yes.
So much about the nature of how the cells are inherent with polymers. Do you need to treat the polymers with any types of polylysine or biomolecules, or is it not necessary?
In most of the cases, yes. For instance, for the patient-derived glioblastoma cells, we had to use a biochemical coating called Caltrex, which is collagen-based coating widely used for patient-derived glioma cells. But also for the work with Jose, for instance, with the PC3 prostate cancer cells, we had to functionalize PEGDA with CNCs in order to promote the adhesion. For neuronal cells, for the microglia, we were very surprised that the primary microglia liked our photopolymer. We actually started to use laminin, and it was actually detrimental in terms of morphology and phenotype development. We found out later, looking at the literature, that laminin is not great for microglia because it can influence some inflammatory responses in the cells themselves. But generally speaking, for instance, the IPS-derived neuronal progenitor cells, we use a combination of polydilysine and laminin, while for the preosteoblast, we use mostly fibronectin. In general, yes. It's true that 2PP companies are developing a lot of biocompatible materials. Nanoscribe is the system that I'm using in my work, and they developed a resin called IPVisio, which is extremely biocompatible. Doesn't need for the glioblastoma cell lines, didn't need any further coating. And the other nice thing is that almost completely non-autofluorescent. That for immunofluorescence is very important.
Two last questions, and then back there. Yeah.
Different cell types in your structures,
Yes, with the slide about the co-culture model with UVEX, human umbilical vein endothelial cells, we combine them with, we pre-culture those cells for 3 days, and then after that we cultured glioblastoma cells for other 2 days, of course, with the with ratios which were taken from literature. And the first results, but they need to be confirmed because it's just one experiment. This is not enough. It seems that UVEX contributed to the resistance of the glioblastoma cells to proton radiation. So in other terms, the number of DNA damage foci of only glioblastoma cells exposed to proton radiation is higher than the number of DNA damage foci of glioblastoma cells in presence of the UVEX, like if they're providing not nutrients, but we have still to understand better what is happening. For the moment, we just have observation, but we have still to think about the interpretation, which is ongoing. So the photoinitiator is not biocompatible at all. Well, most of the photoinitiators are not biocompatible, but as soon as you cross-link them, by two-photon polymerization, you get rid of most of the free radicals. So there is no toxic species around, or at least it is not supposed to be. In order to be completely sure, we always make some so-called detoxification steps. So that means that we have our structures in PBS or even in cell medium, and we wash them like 2-3 times, changing the medium so that we get rid of all toxic species which might be still floating around.
Thank you. Yes, would you like to please take a seat and maybe Dr. Accardo?